Okay, so the last thing that we need to talk about for the chapter 11 are the postsynaptic potentials. And these postsynaptic potentials, remember, that are generated by the neurotransmitter binding to its receptor are graded potentials. And those graded potentials could either depolarize the cell or hyperpolarize the cell. They could be inhibitory or excitatory, and they vary in strength depending on what the neurotransmitter it is, the type of receptor it is, and the time it takes for that neurotransmitter to diffuse across the synaptic cleft and to be degraded. So depending on the effect of the seminal chemical synapse, there's two types of what we call postsynaptic potentials. And again, these are those graded potentials that we're talking about that are occurring in our dendrites and our soma of our neurons themselves. So we have what are called EPSPs, and these are excitatory postsynaptic potentials. And then we have IPSPs, and these are inhibitory postsynaptic potentials. So this is just a little illustration. So basically what's happening is that what I want you to look at here is here's the cell body of one neuron. The cell body of one neuron is in contact with up to hundreds of other of other axon terminals from other neurons coming in. And so all of these different neurons that are synapsing with this one particular neuron are sending different signals. Some of them are excitatory, telling it to fire, and some are inhibitory, telling it not to fire. So how do we decide if we actually do fire the neuron and generate an action potential? So let's talk a little bit more again about the excitatory postsynaptic potentials, or the EPSPs. So anything that would be considered an excitatory potential would be when the neurotransmitter binds to its receptor, which is a chemically gated channel, and allows flow of the ions in opposite directions. However, the flow of sodium coming into the cell is going to be greater than potassium leaving the cell. So we are bringing in more positive charge than we're losing. So that's going to cause a local depolarization. So anytime we bring in something, anytime we have a neurotransmitter that causes us to depolarize the cell by bringing in sodium, that's going to be an excitatory postsynaptic potential, because now we're bringing the cell closer to the threshold of negative 55 millivolts. These excitatory postsynaptic potentials can generate an action potential if they do reach the threshold strength of the negative 55 millivolts, so that it can spread to the axon hillock, which is that point in which our axon is meeting the soma, or the cell body of the neuron, so that we can trigger the opening of those voltage gated channels and then propagate an action potential. So here's just an illustration of what's going on if we are having an excitatory postsynaptic potential. And this particular synapse with one neuron to the next is going to be excitatory. So the receptor right here is going to bind its neurotransmitter and allow much more sodium to come in than potassium's actually leaving. So we're going to get a local depolarization, and that local depolarization is going to bring the potential closer to the threshold right here. In contrast, we could have some synapses in which the neurotransmitter binding to its receptor is going to cause the opposite effect, or hyperpolarization of the cell. And so when the neurotransmitter binds to its receptor, it's going to make the channels open, and one of two channels can open, or two things can happen. If we have a potassium channel open, we're going to move potassium out of the cell, because again, this is going to be a hyperpolarization event in which we're making the cell, the inside of the cell, more negative than it originally was at rest. So if we lose a bunch of positive charge, then we can go from negative 70 to maybe negative 80 millivolts. The other way we can hyperpolarize the cell is by opening chloride ion channels. And because this is a negatively charged ion, we would actually be moving the chloride ions into the cell in order to make the inside of the cell more negative, or to hyperpolarize it itself. So we have two channels and two ions that can move in two different directions in order to create this inhibitory response by by moving the neuron farther away from its threshold. So here's an example. Here's our potassium channel right here. So we can have neurotransmitter bind to the potassium channel, causing us to lose positive charge, which would make the inside of the cell more negative. A neurotransmitter could also bind to a chloride ion channel, causing this channel to open and bringing negative charge into the cell, which is going to also hyperpolarize the cell right there. So if we're looking at it on a graph of a membrane potential, if we started at -70, either one of these channels opening would cause us to have a hyperpolarization, moving us farther away from the threshold needed to generate an action potential. So because it's moving us farther away from the threshold, we say it's an inhibitory postsynaptic potential, or an IPSP. So how do we take all this information? Because like I said, we have a single neuron that has synapses with hundreds of other neurons. Some of these neurons that are synapsing are sending excitatory responses or signals, and some are sending inhibitory. So how do we determine whether or not we actually fire it? Is by what we call summation. So basically, we add up all of the EPSPs, or the excitatory ones, and then we add up all of the IPSPs and the inhibitory ones. And whichever one basically has the most or summates to the highest amount is going to be what that neuron is going to do itself. So a single EPSP can't induce an action potential, but we can add all these excitatory signals coming in from multiple different neurons and add them together or summate them to influence the postsynaptic neuron. And like I mentioned previously, we can also summate or add together IPSPs. So both neurons, or most neurons, are going to receive both signals. So we have to summate all of the incoming signals together. If we have EPSPs that are predominant and bring the membrane to threshold, we can generate an action potential. So we have two types of summation, temporal and spatial. So if we're talking about temporal summation, that's where one or more presynaptic neurons are transmitting impulses in a rapid fire order. So the first impulse is going to produce an excitatory postsynaptic potential, and before it can dissipate, another one is triggered again. So we're kind of adding on to what was already generated. Spatial summation, our postsynaptic neuron is going to be stimulated by a large number of different synapses simultaneously and activate many of the receptors at the same time. And that's going to produce an excitatory postsynaptic potential, which can also add together. So again, we're firing the same one just more frequently before we actually get rid of that graded potential to begin with, and so that we can continue to add. And then the other one is where we use multiple neurons in order to come in and produce the excitatory postsynaptic potentials. So this is just an example. A single EPSP itself can't really generate an action potential, especially if we're sending the signals or firing it fairly slowly. So showing right here, it's going to dissipate with time. So if we wait too long to generate another or fire another one, it's going to have to start basically all over again. In temporal summation, if we're rapidly firing the neuron, the first initial signal is going to come in and cause it to depolarize the cell a little bit. However, before we can hit before the signal can degrade with distance, we fire it again and again and again and again. So each time we fire it, we can basically increase that potential in order to cause an action potential. In spatial summation, we're using multiple signals coming in from multiple neurons, and basically adding those together so that we can generate our action potential and reach our threshold. So this is just showing you here. So if a presynaptic neuron creates an inhibitory one, it can override the excitatory one created by another neuron. So an excitatory postsynaptic potential is going to be right here, bring it closer to threshold. but then we can have an inhibitory one come in and make it go farther back away. Together, they can cancel each other out. So we have synaptic potentiation. So this is something basically, the more we use these neurons or specific neurons within our body, the more we can cause them to excite faster. Okay, it's some sort of like memory and learning process for our neurons. So repeated use of a particular synapse is going to increase the ability of the presynaptic cell to excite the postsynaptic cell. And how does it increase its ability? By increasing the calcium concentration in the presynaptic terminal, and that's going to cause release of more neurotransmitter if we have more calcium coming into the terminal. So that's going to lead to more excitatory postsynaptic potentials, or EPSPs, in the postsynaptic neuron. Potentiation can also cause calcium voltage gated gates to open on the postsynaptic neuron, and then we have some enzymes over here that it can go on and activate to lead to a more effective response. So that's what we call long-term potentiation, and things involved in learning and memory. So basically we train our neurons to fire faster based off of the amount of calcium that we have in the axon terminal and the number of voltage gates we have on the postsynaptic neuron. So we also have postsynaptic inhibition. So release of excitatory neurotransmitter by one neuron can be inhibited by another one if their synapse is on the axoaxonal synapse. So even though these are rare, where we have basically the axon terminal of one axon synapsing right here at the axon, typically at the axon hillock of another one, if one of these, if this neuron is sending an inhibitory signal at this axonal synapse, it can cause the inhibit the release of the neurotransmitter from this neuron to begin with. So this table, this is the last thing I think, yes, that we need to cover in this chapter. This is a really good table just to give you some basic information if any of this is confusing. To compare graded potentials versus action potentials, not only their location, but what it means. So for example here, if we're talking about graded potential, these are only occurring in the cell body and dendrites. Our action potentials are occurring down the length of an axon. The graded potentials only cover short distances because they dissipate with distance. If we're talking about action potentials, these are going to cover long distances down the length of an axon. Graded potentials, they come in various sizes. That's why they're called graded. They do decay with distance. Action potentials are always the same size. We always go from -70 to +30, and they're all or nothing. They either happen or they don't, which means they do not decay with distance. We're talking about how we generate a graded potential. That's going to be from some sort of receptor, either a neurotransmitter receptor or some sort of sensory stimulus like light, pressure, or temperature. Action potentials, the only way we can generate an action potential is by the opening of those voltage gated channels and changing the voltage itself. So graded potentials, we don't have a positive feedback cycle because they decay with distance. Action potentials, we do have a positive feedback cycle. Once we open that first voltage gated channel, we can't stop it. Graded potential repolarization is voltage independent. It occurs when a stimulus is no longer present. If we're talking about repolarization for an action potential, that's voltage regulated. So that's only going to occur once we start inactivating our sodium channels and allowing our voltage gated potassium channels to open right there. Other things, when we talk about differences, summation does not occur at all in action potentials because we're not working with graded potentials at all, just all or nothing responses. We talk about graded potentials, we can summate them or basically add them together. Temporal is basically increasing the frequency of stimuli. By firing it faster, we can reach that threshold to generate an action potential. If we're talking about spatial, we are generating the threshold, the voltage needed for the threshold from multiple sources. So a bunch of different neurons are telling this particular neuron to do the same thing. And then kind of understand the difference when we talk about the function of their signals. These are long distance signals and they are the nerve impulse. And then understand the difference between excitatory and inhibitory postsynaptic potentials. Both of them are short distance. Excitatory is going to depolarize the cell so that we can bring it closer to threshold. Inhibitory is going to hyperpolarize the cell that we can move it farther away from the threshold. Also, you can go in and look back at the channels. Again, we talked about action potentials can only be generated by voltage-gated channels. We have the sodium voltage-gated channel opening first, then the potassium one. For graded potentials, we can have right here a potassium or a sodium channel collectively together in order to basically depolarize the cell or to make it more positive or less negative. We could also have a chemically gated potassium or chloride channel, in which case we'd have the ions moving in different directions depending on which channel would open. Both would be in the goal of hyperpolarizing the cell or making it more negative. So potassium would be leaving the inside of the cell to make it more negative, or chloride ion would be coming into the cell in order to make it more negative. The rest of chapter 11, so sections 9 and 10, are not going to be on the exam. So the remainder of the slides that are in this PowerPoint, you do not need to worry about.